Good day, everyone. So we're reviewing Wetzel's Limnology textbook. This is edition number 4. We're on chapter 15, the phosphorus cycle, and today we'll be looking at stoichiometry of carbon, nitrogen, and phosphorus in particulate organic matter. The elemental chemical composition of planktonic particulate organic matter reflects both the planktonic community structure and biochemical processes, as well as nutrient supply ratios and turnover rates that occurred in the formulation of the composition of the biomass. For example, phosphorus or nitrogen limitation can lead to alterations in the ratios of protein, carbohydrates, and lipid content of cells, as well as the CN ratio. Hence, the CN ratio indicates not only characteristics of the nutrient availability, but also an approximate evaluation of the relative proportions of cellular proteins and non-protein structural elements. A relative constancy in the molar ratio of CNP of 106-16-1 or 7.21 by weight among marine plankton, termed the Redfield ratio, is generally supported by numerous studies. The variance in this ratio is small, usually less than 20%. This constancy has been attributed to the relatively nutrient-sufficient growth conditions of marine plankton and the more homogeneous and stable nature of oceans. In contrast, marked deviations in the cestonic CNP proportions occur in lakes. These particulate composition ratios have been coupled to physiological indicators such as rates of growth and productivity to estimate the nutrient conditions to which phytoplanktons of lakes have been exposed. Such relationships of nutrient stoichiometry can also give insight into species composition, growth rates, and successional patterns of phytoplankton. Elemental cellular stoichiometries of natural phytoplankton communities and the ceston can reflect the type and extent of nutrient limitation and availability. Ratios of elements being loaded to a lake are reflected in the elemental composition of the phytoplankton community. For example, NP ratios in phytoplankton ceston are strongly correlated with NP loading rates to lakes. The CP and NP ratios of lake ceston are generally higher than the Redfield ratios for marine waters. Phosphorus limitation of algal productivity in lakes tends to be much greater than nitrogen limitation, with nitrogen limitation more common in eutrophic lakes because of excessive phosphorus inputs. Streams, shallow lakes, and reservoirs with short residence times have CP ratio less than 350 and NP ratios less than 26, whereas lakes with longer residence times greater than 6 months differentiate from their inflows, typically with CP greater than 400 and NP greater than 30. Tropical lakes tend to have relatively high CN ratios, indicative of potential nitrogen limitation, although the number of lakes sampled was relatively small. Before we move on, let's look at this tables 15-16. Stoichiometric ratios of phytoplankton-dominated cestons of lakes as approximate indicators of relative nutrient limitations. So on the left here, we have the ratio type, we have the deficiency, then the degree of nutrient limitation, none, moderate, and severe. And so this is A is after these authors, and B is comparison of ratio of CN, NP, CP in macromolar per macromole. CCH A, so this is the carbon to chlorophyll A ratios as macromole per macrogram, and physiological ratio of alkaline phosphatase activity APA of chlorophyll A in macromole per macrogram. So they are talking about this right here, the last two lines of this table. But before that, we have all of these ratios here that are macromoles per macromole. So for example, if we looked at the ratio of carbon to nitrogen, the deficiency is nitrogen. And if that ratio is less than 8.3, we don't have any limitation. It's moderate if it's 8.3 to 14.6. I mean by that let's say we have we would have like 10 carbons for 1 nitrogen, so that would give us a value of 10. And in that case, for 10 to 1 ratio, we would have a moderate nutrient limitation for nitrogen. And then when the ratio of C to N is greater than 14.6, we have a severe. Then what is interesting is, for example, if we look at now we jump into the phosphorus-based ratio, so carbon to phosphorus. The deficiency is phosphorus, and so therefore here it takes a lot of carbon to reach that phosphorus deficiency. I mean up to like 258. So greater than 258, we have of a ratio between C and P, we have a severe limitation in nutrients. And then this is carbon to chlorophyll A, and then this is the ratio of alkaline phosphatase activity to chlorophyll A. And so this phosphorus is the limiting factor. Here is general, so I mean maybe like not a specific atom per se, anything. All right, and in here, interestingly enough, they also have a silicon to a phosphorus. So for example, when the silicon to phosphorus ratio is greater than 100, when you have 100 atoms of silicon for 1 atom of phosphorus, you have a severe limitation for silicon in this case, not phosphorus. So silicon is actually the limiting factor. Cellular elemental stoichiometry is affected by the availability of nutrients and therefore can be correlated with the extent of nutrient limitations. Epilimnetic ratios, particularly of NP, are often higher, indicative of high phosphorus demands in proportion to total input than those of the hypolimnion, and CP as well as NP ratios increase with depth. These differences with depth are usually related to high phosphorus use in relation to supply, increased nutrient pool size with depth, and respiration of organic carbon as the system settles. The ratio of fluxes of nutrients into the dissolved nutrient resource pool can markedly influence the community structure of the phytoplankton. As pointed out by Sterner, external sources of nutrient loading can be and have been evaluated with some degree of accuracy, but rates and controls of internal regeneration of nutrients by physical and biological processes are much less quantified to levels that allow effective use of predictive models. For example, denitrification was the most important mechanism for reducing NP ratios in the water column, whereas both nitrogen fixation and sediment resuspension raised NP ratios. The sediments tend to be the major nitrogen source, particularly in littoral areas and anoxic zones. The NP and Si to P supply ratios are fundamental axes along which structures of phytoplanktonic communities vary. Diatoms and some chrysophyte algae have absolute silica requirements. Although total phytoplankton biomass is not limited by silica availability, the phytoplankton community composition, interspecific competition, and succession can be altered markedly. Species of diatoms with low Si to P ratio requirements, for example, often develop maximally during seasonal periods of lowest silicon to phosphorus ratios. In another example, many cyanobacteria are capable of nitrogen fixation, which allows the species to maintain high growth rates in habitat low in available combined inorganic nitrogen, where adequate supply of trace metals needed for nitrogen fixation and heterosynthesis are available. The relative competitive capacities of and proportions of cyanobacteria in epilimnetic phytoplankton communities are thus indicated by the NP ratio. However, cyanobacteria tend to be rare at NP greater than 29 by weight. Zooplankton and larger animals usually constitute small portion of the total dissolved ancestor pools of nitrogen and phosphorus. For example, in an alpine lake in California, zooplankton always constituted less than 5% and less than 10% of the pelagic storage compartments for nitrogen and phosphorus, respectively. However, during brief periods of high zooplankton biomass, the zooplanktonic contribution to total cystonic elemental stoichiometry can be significant. Although the NP and CP ratios do not vary greatly intraspecifically among zooplankton, considerable differences occur interspecifically. Zooplankton NP ratios may be altered by the NP ratios of cestone as certain individual species populations of zooplankton which grow and reproduce most efficiently at a given NP supply ratio come to dominate the zooplankton community because the biochemical requirements of crustacean zooplankton for an NP ratios in their tissue is below the ratio of most cystonic assemblages. There is a tendency for crustacean zooplankton to retain phosphorus preferentially to nitrogen. As a result of this process, nutrient regeneration via feces of zooplankton can be skewed toward nitrogen and thereby enhance phosphorus limitation to phytoplankton during short periods of intense cladocerian grazing. In examining the chemical composition of particulate organic matter, large zooplankton can be effectively separated from smaller particles. However, detrital particles nearly always dominate between 50 and 80% over algal and bacterial biomass in the remaining cestone. If the planktonic microbiota are reproducing rapidly, much of the detritus present can have chemical composition relatively similar to that of the living plankton. Appreciable loadings of particulate organic matter from the structural tissues of terrestrials and littoral plants, however, can result in cestone relatively rich in carbon and lesser amounts of nitrogen and lead to high CN, CP, and NP ratios. Although much of the particulate organic matter from external sources tend to be deposited in nearshore regions, in natural lakes, importation from the drainage basin is much greater in the reservoir ecosystems. In addition, because of the high ratio of drainage basin area to reservoir area, large quantities of inorganic particles, for example clay, can be imported to the pelagic zone. These inorganic particles of the subsection can contain high and variable quantities of nutrients that are undifferentiated from particulate organic matter of biota. It can lead to skewed results requiring care and interpretation. However, in most lakes, with water residence time of several months or longer, cystonic composition ratios reflect the general status of the availability of nitrogen and phosphorus for planktonic growth in these waters. Furthermore, the predictive value of nutrients present in the cestone as a reflection of nutrient availability has been extended by the obvious relationship of energy available as light for photosynthesis to nutrient availability when incorporated into the cestone. Phosphorus availability is low. CP ratios of particulate organic matter will tend to be low because photosynthetic productivity is low. Differences in nutrient use efficiency may occur at different light availability, and these ratios will be translated to higher trophic levels feeding upon them. Low light phosphorus ratios of cestone likely indicate simultaneous carbon and organic matter limitation in other dependent trophic levels, whereas high light phosphorus ratios suggest phosphorus limitation among several dependent trophic levels. Effects of decreasing phosphorus on productivity. The reduction of the productivity of lakes by decreasing phosphorus loading can be very effective. Many examples are given in reviews by Voltweider and Chandler. Among the most frequently discussed example is Lake Washington. Lake Washington was enriched with increasing volume of effluents from secondary storage treatment facility during the period from 1941 to 1953. Production increased markedly and the phytoplankton became more abundant. Phosphate concentration also increased proportionally, much more than those of nitrate or carbon dioxide. Effluent was diverted away from the lake in 1963, and by 1969, phosphate had decreased to 28% of its 1963 value. Summer chlorophyll A concentration had decreased about as much, but nitrate and total CO2 fluctuated from year to year at a relatively high values. Reduction in phytoplankton and phosphorus continued to a relatively stable level in the 1990s. A very similar situation occurred in Gravenhurst Bay, which with tertiary treatment of wastewater resulting in the same improvement as wastewater diversion in the Lake Washington case. The rate at which the productivity of a lake will revert toward conditions existing prior to the increased phosphorus loading is variable and depend upon basin morphometry, water chemistry, the nature of the phosphorus sources, and the rate of internal phosphorus loading. The loading of lakes with nitrogen and phosphorus is further influenced by the ratio of the surface area of the drainage basin to that of the lake. Depending on the percentage losses from the land, some of which may be under cultivation, loading increases roughly in proportion to this ratio. Upon reduction of loading rates to or below those of the lake prior to accelerated inputs, recovery will require 2 to 10 years for lakes of average size and average hydrological replenishment time. The relative residence time, or inversely the flow-through rate of phosphorus, is directly coupled to the efficiency of sediment removal and retention capacities in the regulation of phytoplankton. Whole lake experiment at the experimental lake area in northwestern Ontario show that oligotrophic lakes will respond rapidly to large increases in loads. In contrast, eutrophic lakes with a long eutrophication history and large accumulation of nutrients in the sediment will respond slowly to decreasing loads because of the continued phosphorus release from anaerobic sediments, internal loading. The rate of recovery is much slower in shallow lakes, while the internal aerial loading of phosphorus from sediment sources is high related to lake volume. In eutrophic Shagawa Lake, for example, where external phosphorus loading was reduced by greater than 70%, predicted reduced equilibrium levels were not achieved. Shagawa Lake is shallow, has an anaerobic hypolimnion, and has an extensively developed littoral zone of submerged macrophytes, all of which contribute to the enhanced release of sediment phosphorus. Sediment phosphorus is the main source of phosphorus for macrophytes, which can release phosphorus to the water column when they senesce. Internal loading from sediments phosphorus sources has been observed in many other shallower lakes, reservoirs, or lakes with anoxic hypolimnia. The magnitude of internal phosphorus loading can be predicted from the product of an average rate of phosphorus release from anoxic sediments, the surface area of the anoxic sediment, and the period of anoxia. Such internal loadings slows the rate of lake recovery implemented by the reduction of phosphorus loading from external allochthonous sources. In most lakes, however, the internal loading contribution is a relatively small portion of the external sources. These authors so that regulations of loading waste can often be effective in controlling phytoplankton productivity. I think that's going to be it for today, and tomorrow we'll pick it up with this area here related to the retention capacity. Thank you for tuning in, and I'll see you tomorrow. Bye.